The modern global food supply chain is designed for peak efficiency, moving produce across the planet with surgical precision. Yet, a single soil-borne fungus is currently threatening to dismantle one of its most critical components. Every year, global banana production exceeds 120 million tons. The vast majority of this fruit grows on industrial plantations, arranged in identical rows that prioritize high yields and ease of management. Of the millions of tons exported for international trade, the Cavendish variety accounts for between 95 and 99%. Moving this specific volume requires a massive, highly specialized shipping infrastructure. This visible abundance at the supermarket masks a deep systemic vulnerability. The entire global trade depends on a strategy of extreme genetic uniformity. Commercial Cavendish bananas are genetically identical clones. They are sterile plants, grown from tissue cultures in laboratories before being moved to the field, ensuring every plant in a 1,000,000-hectare plantation is a biological twin of the next. Cloning offers distinct industrial advantages. It provides consistent bunch sizes, predictable ripening times, and skins durable enough to withstand weeks of ocean transport. This uniformity created a perfect opening for Fusarium oxysporum tropical race 4, a soil-borne fungal pathogen specifically adapted to bypass the Cavendish's defenses. While genetic variation in a wild forest creates natural barriers to disease, a monoculture offers a clear path. Once the fungus adapts to the specific weaknesses of one plant, it effectively has access to the entire identical population worldwide. The very traits that made the Cavendish an industrial success are the same traits that left it biologically defenseless against TR4. TR4 is a vascular wilt pathogen. It enters through microscopic wounds in the roots and colonizes the xylem, the plant's internal plumbing, eventually blocking the transport of water and nutrients. The infection is difficult to spot. Plants remain asymptomatic for up to 2 years, allowing the fungus to spread undetected before any wilting appears. To survive dormant periods, the pathogen produces chlamydospores, thick-walled structures built for long-term persistence. These spores can survive in empty soil for over 40 years. There is no known chemical cure or soil fumigant that can eradicate them once established. TR4 does more than kill a single harvest. It permanently alters the land, making it unsuitable for susceptible varieties for decades. Emerging in Taiwan in 1967, TR4 spread across Southeast Asia, hitchhiking globally in contaminated soil on boots, shared machinery, and shipping containers. It crossed oceans. It breached Latin America's export heartland in Colombia in 2019, reaching Peru in 2021 and Venezuela in 2023. By late 2025, the fungus reached Ecuador, the source of 1/3 of the world's exported bananas. This pattern follows a historical precedent. In the mid-20th century, an earlier race of this same fungus decimated the Gros Michel banana, forcing the industry to pivot to the Cavendish for its resistance at the time. Global agriculture has essentially repeated the same mistake, replacing one vulnerable monoculture with another while maintaining the same underlying structure. Current projections estimate hundreds of billions of dollars in global economic losses as TR4 spreads. Supply shocks in major export hubs are expected to drive consumer prices higher worldwide. For millions of people in tropical regions, these crops are essential dietary staples. In heavily affected areas, the loss of production threatens both local caloric intake and the primary livelihoods of smallholder farmers. The Cavendish crisis is a case study for other global staples. Crops like wheat, rice, and maize also rely on narrow genetic bases, optimized for industrial scale, leaving them exposed to similar evolutionary pressures. Industrial farming has successfully delivered cheap calories, but it has done so by removing the biological diversity that traditionally provided systemic resilience. Immediate containment requires rigorous biosecurity. Plantations must implement strict disinfection protocols for vehicles and equipment while ensuring that only certified clean tissue cultures are used for new plantings. Scientists are also working to introduce resistance through gene editing and traditional crossbreeding with wild banana varieties. These efforts aim to preserve the traits consumers expect while upgrading the plant's internal defenses. The long-term solution involves moving away from massive, contiguous monocultures. Reintroducing crop diversification and multivarietal systems creates a more complex landscape that is naturally harder for a single pathogen to exploit. The TR4 crisis reveals the high cost of prioritizing short-term efficiency over biological stability. Our food security depends on moving past the assumption that genetic uniformity can outpace the constant speed of evolution.